Biology or life science, meiosis, pronounced meiosis, is a process of reduction division in which a number of chromosomes per cell is cut in half. In animals, meiosis always results in the formation of gametes. The word meiosis comes from the Greek verb mein, meaning to make small, since it results in a reduction in chromosome number in the gamete cell. Meiosis is essential for sexual reproduction and therefore occurs in all eukaryotes, including single-celled organisms that reproduce sexually. Few eukaryotes, notably the beteloid rotifers, have lost the ability to carry out meiosis and have acquired the ability to reproduce by parthenogenesis. Meiosis does not occur in archaea or bacteria, which reproduce via asexual processes such as mitosis or binary fission. Each cell has half the number of chromosomes as the parent cell. During meiosis of a diploid germ cell, which is composed of long segments of DNA packaged into chromosomes, undergoes DNA replication followed by 2 rounds of division, resulting in 4 haploid cells. Each of these cells contain one complete set of chromosomes, or half of the genetic content of the original cell. If meiosis produces gametes, these cells must fuse during fertilization to create a new diploid cell, or zygote before any new growth can occur. Thus, the division mechanism of meiosis is a reciprocal process to the joining of 2 genomes that occurs at fertilization. As the chromosomes of each parent undergo genetic recombination during meiosis, each gamete, and thus each zygote, will have a unique genetic blueprint encoded in its DNA. Together, meiosis and fertilization constitute sexuality in the eukaryotes, and generate genetically distinct individuals in populations. In plants, and in many protists, meiosis results in the formation of haploid cells that can divide vegetatively without undergoing fertilization. In these groups, gametes are produced by mitosis. Meiosis uses many of the same biochemical mechanisms employed during mitosis to accomplish the redistribution of chromosomes. There are several features unique to meiosis, most importantly the pairing and genetic recombination between homologous chromosomes. History. Meiosis was discovered and described for the first time in sea urchin eggs in 1876, by noted German biologist Oskar Hertwig, 1849–1922. It was described again in 1883, at the level of chromosomes, by Belgian zoologist Edouard van Benden, 1846–1910, in Ascaris worms' eggs. The significant meiosis reproduction and inheritance, however, was described only in 1890 by German biologist August Weismann, 1834–1914, who noted that 2 cell divisions were necessary to transform one diploid cell into 4 haploid cells if the number of chromosomes had to be maintained. In 1911, the American geneticist Thomas Hunt Morgan, 1866–1945, observed crossover in Drosophila melanogaster meiosis and provided the first true genetics. Meiosis is thought to have appeared 1.4 billion years ago. The only supergroup of eukaryotes which does not have meiosis in all organisms is Excavata. The major supergroups, Opisthokonts, Amoebozoa, Rosaria, Archaeoplastida and Chromalveolates all seem to have genes for meiosis universally present, even if not always functional. Some Excavata species do have meiosis, which is consistent with the hypothesis that Excavata is an ancient, paraphyletic grade. An example of eukaryotic organism in which meiosis does not exist is euglenoid. Occurrence of meiosis in eukaryotic life cycles. Meiosis occur in eukaryotic life cycles involving sexual reproduction, comprising of the constant cyclical process of meiosis and fertilization. This takes place alongside normal meiotic cell division. In multicellular organisms, there is an intermediary step between the diploid and haploid transition where the organism grows. The organism will then produce the germ cells that continue in the life cycle. The rest of the cells, called somatic cells, function within the organism and will die with it. Cycling meiosis and fertilization events produces a series of transitions back and forth between alternating haploid and diploid states. The organism cycle can occur either during the diploid state, gametic life cycle, or during the haploid state, zygotic life cycle, or both, sporic life cycle, in which there are 2 distinct organism phases, one during the haploid state and the other during the diploid state. In this sense, there are 3 types of life cycles that utilize sexual reproduction, differentiated by the location of the organism's phases. In the gametic life cycle, the species is diploid, grown from a diploid cell called the zygote. In the zygotic life cycle, the species is haploid instead, spawned by the proliferation and differentiation of a single haploid cell called the gamete. Humans, for example, are diploid creatures. Human stem cells undergo meiosis to create haploid gametes, which are spermatozoa for males or ova for females. These gametes then fertilize in the fallopian tubes of the female, producing a diploid zygote. The zygote undergoes progressive stages of mitosis and differentiation, turns into a blastocyst and then gets implanted in the uterus endometrium to create an embryo. In the gametic life cycle, of which humans are a part, the living organism is diploid in nature. Here, we will generalize the example of human reproduction stated previously. The organism's diploid germline stem cells undergo meiosis to create haploid gametes, which fertilize to form a zygote. The diploid zygote undergoes repeated cellular division by mitosis to grow into the organism. Mitosis is a related process to meiosis that creates 2 cells that are genetically identical to the parent cell. The general principle is that mitosis creates somatic cells and meiosis creates germ cells. In the zygotic life cycle, the living organism is haploid. 2 organisms of opposing gender contribute their haploid germ cells to form a diploid zygote. The zygote undergoes meiosis immediately, creating 4 haploid cells. These cells undergo mitosis to create the organism. Many fungi and many protozoa are members of the zygotic life cycle. Finally, in the sporic life cycle, the living organism alternates between haploid and diploid states. Consequently, this cycle is also known as the alternation of generations. The diploid organism's germline cells undergo meiosis to produce gametes. The gametes proliferate by mitosis, growing into a haploid organism. The haploid organism's germ cells then combine with another haploid organism's cells, creating the zygote. The zygote undergoes repeated mitosis and differentiation to become the diploid organism again. The sporic life cycle can be considered a fusion of the gametic and zygotic life cycles. Thus, because meiosis is a one-way process, it cannot be said to engage in a cell cycle as mitosis does. However, the preparatory steps that lead up to meiosis are identical in pattern and name to the interphase of the meiotic cell cycle. Interphase is divided into 3 phases. Growth 1 G1 phase immediately follows cytokinesis. This is a very active period, where the cell synthesizes its vast array of proteins, including the enzymes and structural proteins it will need for growth. In G1 stage, each of the 46 human chromosomes consists of a single, very long, molecule of DNA. At this point, cells are 46, 2n, identical to somatic cells. Synthesis S phase. The genetic material is replicated, each of its chromosomes duplicates 46, 2n. The cell is still diploid, however, because it still contains the same number of centromeres. However, the identical sister chromatids are in the chromatin form because spiralization and condensation into denser chromosomes have not taken place yet. It will take place in prophase I in meiosis. Growth 2 G2 phase. G2 phase is absent in meiosis. Interphase is immediately followed by meiosis I in meiosis II. Meiosis I consists of segregating the homologous chromosomes from each other, then dividing the diploid cell into 2 haploid cells each containing one of the segregates. Meiosis II consists of decoupling each chromosome's sister strands, chromatids, segregating the DNA into 2 sets of strands, each set containing one of each homolog, and dividing both haploid, duplicated cells to produce 4 haploid, unduplicated cells. Meiosis I and II are both divided into prophase, metaphase, anaphase, and telophase subphases, similar in purpose to their analogous subphases in the meiotic cell cycle. Therefore, meiosis encompasses the interphase G1, S, G2, meiosis I, prophase I, metaphase I, anaphase I, telophase I, and meiosis II, prophase II, metaphase II, anaphase II, telophase II. Meiosis generates genetic diversity in 2 ways. 1. Independent assortment of chromosomes at both of the meiotic divisions allows genetic differences among gametes, and 2. Physical exchange of chromosomal regions by homologous recombination during prophase I results in new genetic combinations within chromosomes. Meiosis I. In meiosis I, the homologous pairs in a diploid cell separate, producing 2 haploid cells 46, n. The 46 chromosomes number is significant. A regular diploid cell contains 46 chromosomes and is considered 2n because it contains 23 pairs of homologous chromosomes. However, after meiosis I, although the cell contains 46 chromosomes, it is only considered n because later in anaphase I the identical sister chromatids will remain together as the spindle pulls the pair toward the pole of the new cell. In II, a process similar to mitosis will occur whereby the sister chromatids are finally split, creating 2 haploid cells 23, n. Prophase I. Homologous chromosomes pair and crossing over, or recombination, occurs, a step unique to meiosis. Chromosomes form structures called synapses. Chromosomes are called bivalents or tetrads, which have 2 chromosomes and 4 chromatids, with 1 chromosome coming from each parent. At this stage, non-sister chromatids may cross over at points called chiasmata. Leptidine. The first stage of prophase I is the leptidine stage, also known as leptoma, from Greek words meaning thin threads. During this stage, individual chromosomes begin to condense into long strands within the nucleus. However, the 2 sister chromatids are still so tightly bound that they are indistinguishable from one another. Chromosomes in the leptidine stage show a specific arrangement where the telomeres are oriented towards the nuclear membrane. Hence, this stage is called bouquet stage. Zygodyne. The zygodyne stage, also known as zygoma, from Greek words meaning paired threads, occurs as the chromosomes approximately line up with each other into homologous chromosomes. The combined homologous chromosomes are said to be bivalent. They may also be referred to as a tetrad, a reference to the 4 sister chromatids. The 2 homologous chromosomes become zipped together, forming the synaptonemal complex, in a process known as synapsis. The pachydyne stage, also known as pachyma, from Greek words meaning thick threads, contains the following chromosomal crossover. Non-sister chromatids of homologous chromosomes randomly exchange segments of genetic information over regions of homology. Sex chromosomes, however, are not identical, and only exchange information over a small region of homology. Exchange takes place at sites where recombination nodules or chiasmata, singular, chasma, have formed. The exchange of information between the non-sister chromatids results in a recombination of information. Each chromosome has the complete set of information it had before, and there are no gaps formed as a result of the process. Because the chromosomes cannot be distinguished in the synaptonemal complex, the actual act of crossing over is not perceivable through the microscope. During the diploidine stage, also known as diploma, from Greek words meaning 2 threads, the synaptonemal complex degrades and homologous chromosomes separate from one another a little. The chromosomes themselves uncoil a bit, allowing some transcription of DNA. However, the homologous chromosomes of each bivalent remain tightly bound at chiasmata, the regions where crossing over occurred. The chiasmata remain on the chromosomes until they are severed in anaphase I. In fetal oogenesis, all developing oocytes develop to this stage and stop before birth. This suspended state is referred to as the dictyoten stage and remains so until puberty. In males, only spermatogonia exist until meiosis begins at puberty. Diakinesis. Chromosomes condense further during the diakinesis stage, from Greek words meaning moving through. This is the first point in meiosis where the 4 parts of the tetrads are actually visible. Sites of crossing over untangle together, effectively overlapping, making chiasmata clearly visible. As observation, the rest of the stage closely resembles prometaphase of mitosis. The nucleoli disappear, the nuclear membrane disintegrates into vesicles, and the meiotic spindle begins to form. Synchronous processes. During these stages, centrosomes, each containing a pair of centrioles, are migrating to the 2 poles of the cell. These cells, which were duplicated during S phase, function as microtubule organizing centers nucleating microtubules, essentially cellular ropes and poles, during crossing over. They invade the nuclear membrane after it disintegrates, attaching to the chromosomes at the kinetochore. The kinetochore functions as a motor, pulling the chromosome along the attached microtubule toward the originating centriole, like a train on a track. There are kinetochores on each tetrad, but the pair of kinetochores on each sister chromatid fuses and functions as a unit during meiosis I. Microtubules that attach to the kinetochores are known as kinetochore microtubules. Other microtubules will interact with microtubules from the opposite centriole. These are also non-kinetochore microtubules. Metaphase I. Homologous pairs move together with plate. As kinetochore microtubules from both centrioles attach to their respective kinetochores, the homologous chromosomes align along an equatorial plane that bisects the spindle, due to continuous counterbalancing forces exerted on the bivalence by the microtubules emanating from the two kinetochores of homologous chromosomes. The physical basis of the independent assortment of chromosomes is the random orientation of each bivalent along the metaphase plate. Anaphase I. Kinetochore microtubules shorten, severing the recombination nodules and pulling homologous chromosomes apart. Since each chromosome only has one functional unit of a pair of kinetochores, three whole chromosomes are pulled toward opposing poles, forming two haploid sets. Each chromosome still contains a pair of sister chromatids. Non-kinetochore microtubules lengthen, pushing the centrioles further apart. The cell elongates in preparation for division down the middle. Telophase I. The last meiotic division effectively ends when the centromeres arrive at the poles. Each daughter cell now has half the number of chromosomes, but each chromosome consists of a pair of chromatids. This effect produces a variety of responses from the neurosyncromatic enzyme, also known as NSE. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid set. The chromosomes uncoil back into chromatin. Cytokinesis, the pinching of the cell membrane in animal cells or the formation of the cell wall in plant cells, occurs, completing the creation of two daughter cells. Cells enter a period of rest known as interkinesis or interphase II. No DNA replication occurs during this stage. Telophase I contains no nucleus, two daughter cells, and chromatids remain attached. Meiosis II. Meiosis II is the second part of the meiotic process. The process is similar to mitosis and meiosis I. End result is production of four haploid cells, 23, one end from the two haploid cells, 46, one end produced in meiosis I. Prophase takes an inversely proportional time compared to telophase I. In this prophase we see the disappearance of the nucleoli in the nuclear envelope again, as well as the shortening and thickening of the chromatids. Centrioles move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase II, the centromeres contain two kinetochores that attach to spindle fibers from the centrosomes centrioles at each pole. The new equatorial metaphase plate is rotated by 90 degrees when compared to meiosis I, perpendicular to the previous plate. This is followed by anaphase II, where the centromeres are cleaved, allowing microtubules attached to the kinetochores to pull the sister chromatids apart. Sister chromatids by convention are now called sister chromosomes as they move toward opposing poles. Process ends with telophase II, which is similar to telophase I, and is marked by uncoiling and lengthening of the chromosomes and the disappearance of the microtubules. Nuclear envelopes reform and cleavage or cell wall formation eventually produces a total of four daughter cells, each with a haploid set of chromosomes. Meiosis is now complete. The significance of meiosis. Meiosis facilitates stable sexual reproduction. Without the having of ploidy, or chromosome count, fertilization would result in zygotes that have twice the number of chromosomes than the zygotes from the previous generation. 6 generations would have an exponential increase in chromosome count, resulting in an unwieldy genome that would cripple the reproductive fitness of the species. Polyploidy, the state of having 3 or more sets of chromosomes, also results in developmental abnormalities or lethality. Polyploidy is poorly tolerated in animal species. Plants, however, regularly produce fertile, viable polyploids. Polyploidy has been implicated as an important mechanism in plant speciation. Most importantly, however, meiosis produces genetic variety in gametes that propagate to offspring. Recombination and independent assortment allow for a greater diversity of genotypes in the population. As a system of creating diversity, meiosis allows a species to maintain stability under environmental changes. Non-disjunction. The normal separation of chromosomes in meiosis I or sister chromatids in meiosis II is termed disjunction. When the separation is not normal, it is called non-disjunction. This results in the production of gametes which have either more or less of the usual amount of genetic material, and is a common mechanism for trisomy or monosomy. Non-disjunction can occur in the meiosis I or meiosis II phases of cellular reproduction, or during mitosis. This is a cause of several medical conditions in humans. Down syndrome, trisomy of chromosome 21. Patau syndrome, trisomy of chromosome 13. Edwards syndrome, trisomy of chromosome 18. Klinefelter syndrome, extra X chromosomes in males, i.e. XXY, XXXY, XXXXY. Turner syndrome, atypical X chromosome dosage in females, i.e. XO, XXX, 4X. XYY syndrome, an extra Y chromosome in males. Meiosis in humans. In females, meiosis occurs in cells known as oogonia, singular, oogonium. Each oogonium that initiates meiosis will divide twice to form a single oocyte and 3 polar bodies. However, before these divisions occur, these cells stop at the diplodene stage of meiosis I and lay dormant within a protective shell of somatic cells called the follicle. Follicles begin growth at a steady pace in a process known as folliculogenesis, and a small number enter the menstrual cycle. Menstruated oocytes continue meiosis I and arrest at meiosis II until fertilization. The process of meiosis in females occurs during oogenesis, and differs from the typical meiosis in that it features a long period of meiotic arrest known as the Dichtat stage and lacks the assistance of centrosomes. In males, meiosis occurs in precursor cells known as spermatogonia that divide twice to become sperm. These cells continuously divide without arrest in the seminiferous tubules of the testicles. Sperm is produced at a steady pace. The process of meiosis in males occurs during spermatogenesis. D3 cell cycle. Fundamental concepts of cell cycle. The cell cycle is the sequence of events between successive cell divisions. Many different processes must be coordinated during the cell cycle, some of which occur continuously, e.g. cell growth, and some discontinuously, as events are landmarks, e.g. cell division. Cell division must be coordinated with growth and DNA replication so that cell size and DNA content remain constant. The cell comprises a nuclear or chromosomal cycle, DNA replication and partition, and a cytoplasmic or cell division cycle, doubling and division of cytoplasmic components, which in eukaryotes includes the organelles. The DNA is considered separately from other cell contents because it is usually present in only one or two copies per vegetative cell, and its replication and segregation must therefore be precisely controlled. Most of the remainder of the cell contents are synthesized continuously and in sufficient quantity to be distributed equally into the daughter cells when the parental cell is big enough to divide. An centrosome, an organelle that is pivotal in the process of chromosome segregation itself, which is duplicated prior to mitosis and segregated into the daughter cells with the chromosomes, the centrosome cycle. In eukaryotes, the two major events of the chromosomal cycle, replication and mitosis, are controlled so that they can never occur simultaneously. Conversely, in bacteria the analogous processes, replication and partition, are coordinated so that partially replicated chromosomes can segregate during rapid growth. The eukaryotic cell cycle is divided into discrete phases which proceed in a particular order, whereas the stages of the bacterial cell cycle may overlap. The progress of the eukaryotic cell cycle is controlled at checkpoints where regulatory proteins receive input from monitors of the cell cycle itself, intrinsic information, and monitors of the environment, extrinsic information. Intrinsic monitoring ensures that the stages of the cell cycle proceed in the correct order and that one stage is completed before the next begins. Extrinsic monitoring coordinates cell division with cell growth and arrests the cell cycle if the environment is unsuitable. The cell cycle is controlled by protein kinases. Cell cycle transitions involve positive feedback loops which cause sudden bursts of kinase activity, allowing switches in the states of phosphorylation of batteries of effector proteins. Cell cycle checkpoints are regulatory systems which inhibit those kinases if the internal or external environment is unsuitable. The alternation of DNA replication and mitosis is controlled by negative feedback. Mitosis is inhibited by unfinished DNA replication, and DNA replication is prevented during mitosis by the phosphorylation and inactivation of a protein required for replication. The cell cycle is the result of a complex network of information, in which kinases are controlled by the integration of multiple positive and negative signals. 1. The bacterial cell cycle. DNA replication and growth coordination. The model, or I+C+D model, divides the bacterial chromosome cycle into three phases: the interval phase, the chromosome replication phase and the division phase, represented by the letters I, C and D, respectively. DNA replication occurs during the C phase. Its duration is fixed, about 40 minutes in E. coli, reflecting the time taken to replicate the whole chromosome. The D phase begins when replication is complete, and culminates in cell division. The D phase is also fixed, about 20 minutes in E. coli and can be regarded as the time required to synthesize the cellular components required for cell division. The minimum duration of the chromosome cycle in E. coli is thus 1H. The C+D is fixed. Any change in the cell doubling time must reflect a change in the duration of I, the interval between successive initiations of replication. The doubling time of E. coli can be as long as 3H or as short as 20 minutes. During slow growth, I greater than C+D and replication is completed before cell division. During rapid growth, however, the doubling time is shorter than the time taken to complete a round of replication and cell division. The only way that can accommodate its fixed chromosome cycle into the accelerated cell division cycle is to make I less than C+D, i.e. new rounds of replication must begin before the previous round is complete. Therefore, rapid growth, daughter cells inherit chromosomes which are already partially replicated, multiforked osomes, so that replication can be completed before the next round of cell division. The frequency of initiation is thought to be controlled by a positive regulator which must be present at a certain critical concentration per origin of replication Q, versus for initiation to be successful. During rapid growth, the regulator accumulates more quickly, allowing more frequent initiation. Once initiation has occurred, the number of origins in the cell doubles and the effective concentration of the regulator is halved so that it must accumulate again for another round of initiation to occur. The existence of a positive regulator is predicted because de novo protein synthesis is required for initiation. However, the nature of this putative molecule is unknown. The replicator protein DNA is a possible candidate, and factors which control methylation at ORI-C and the DNA promoter could also be involved. Q. V. Origin of replication, deamethylation. Partition and cytokinesis. The partition of the replicated chromosome marks the culmination of the chromosome cycle and is followed by cell division. A septum forms at the midpoint of the parental cell, which is identified by a paraseptal annulus, a region of modified cell envelope where the inner and outer membranes are joined together around the circumference of the cell. Additional annuli form by duplication and migrate to positions equivalent to 1/4 and 3/4 cell lengths, and these are the sites of septation in daughter cells during the next round of cell division. Once the septum has formed, the cell undergoes cytokinesis. It divides by binary fission. The identification of mutants which disrupt cell division or partitioning has shown that the two processes can be unhitched, and such mutants fall into several categories. FTS mutants are deficient in septum formation and thus form filaments that are temperature sensitive, hence the name. The filaments often contain regularly spaced nucleoids, indicating that replication and partitioning mechanisms are still functioning normally. Mutants generate septa too frequently, resulting in the formation of minicelles, which are small cells which contain no chromosomal DNA, although they may contain plasmids. Finally, pure mutants form normal-sized cells but fail to partition the chromosomes properly, so that diploid and anucleate cells arise with high frequency. Pathway controlling cell division and partition has yet to be determined in full, but several key players have been identified. A good candidate for the initiator of cell division is FTSC. The protein is structurally and functionally similar to tubulin, which forms the contractile ring in eukaryotic cells. It is distributed ubiquitously during most of the cell cycle, but is localized around the annulus at the beginning of the D phase as AZ ring. The bins appears to correlate exactly with the frequency of cell division. The septs mutants fail to form septa and generate filaments, whereas overexpression causes the production of too many septa, and hence minicelles. The zipaprotein may be important for the localization of FTSC because its N-terminus is membrane-associated and its C-terminus interacts with FTSC. This septum is positioned in the cell, although nucleation sites probably exist because filaments resulting from temperature-sensitive FTSC mutations rapidly form contractile rings at regular intervals when shifted to the permissive temperature. G. The Min-B locus limits septation to the central annulus and suppress the process at the terminal annuli remaining from previous cell divisions. Zinc and Min are septation inhibitors, whereas Min antagonizes Min-CD activity. The correct balance of all three products thus inhibits terminal septation but protects the central annulus from inhibition. The partition of bacterial chromosomes proceeds similarly to plasmid partition Q, V. Partitioning may be the association of the chromosome with the cell membrane, and may be regulated by replication. The origin and terminus of replication, as well as active replication forks, are membrane-associated. Bacterial defect partition fall into two categories: those which interfere with the separation of interlocked replicated chromosomes, these include topoisomerase and exor-site-specific recombinase mutants, and those which affect the partition process itself. In the latter category, cis-acting sites have been found on the chromosome, but several trans-acting factors have been identified, e.g. the membrane protein MUPA and the microtubule-associated protein MUC-B. Mutations in both genes generate anucleate cells. 2.1 The eukaryotic cell cycle. The standard eukaryotic cell cycle. The standard eukaryotic cell cycle is divided into 4 non-overlapping phases. The chromosome cycle, DNA synthesis and mitosis, occur during the S phase and the M phase, respectively, and in most cell cycles these landmarks are separated by G1 and G2 gap phases, during which mRNAs and proteins accumulate continuously. Figure 2.1. Process of crossing from one phase of the cell cycle to the next is a cell cycle transition. Where mitosis is a dramatic event that involves visible reorganization of cell structure, the rest of the cell cycle is unremarkable to the eye and is termed the interphase. Variation theme table 2.2 includes cell cycles where one or both gap phases are omitted, or where either the S phase or the M phase is omitted, leading to having or doubling of the DNA content, respectively. Addition, a cell may be arrested, indefinitely or permanently delayed, at any stage of the cell cycle, as occurs during oocyte maturation and in postmitotic cells such as neurons. Animal cells may withdraw from a cell cycle altogether, entering the quiescent state termed GO, where both growth and division are repressed. Selecting requirement for growth factors and other signaling molecules in the environment, and imposes an extra level of regulation on the cell cycle so that the growth and division of individual cells can be coordinated in the context of a multicellular organism. The abnormal cell proliferation seen in cancer is caused by the failure of this regulatory mechanism. Cell cycle checkpoints. The prime function of the cell cycle is to duplicate the genome precisely and divide it equally between two daughter cells. For this reason, it is important that the events of the cell cycle proceed in the correct order, and that each stage of the cell cycle is complete before the next commences. DNA remains constant only if DNA replication alternates with mitosis, if mitosis occurs after the completion of DNA replication, and replication commences after mitosis has precisely divided the DNA. The cell meets these criteria by organizing the cell cycle as a dependent series of events. Thus, mitosis is blocked, the cell cycle arrests at the M phase until the block is removed. It does not go ahead and replicate the DNA anyway, i.e. DNA replication is dependent upon the completion of mitosis. Similarly, if DNA replication is prevented, the cell does not attempt to undergo mitosis, because mitosis is dependent upon the completion of DNA replication. Another function of the cell cycle is to coordinate the chromosome cycle with cell growth, so there is no progressive loss or gain of cytoplasm, and no cell proliferation in an unsuitable environment. The cell cycle is thus also dependent upon cell size and is regulated by nutrient availability, the presence of mating pheromones in yeast, and the presence of growth factors and hormones in animals. The cell possesses a number of regulatory systems which can sense the progress of the cell cycle and can inhibit subsequent stages in the event of failure. These regulatory mechanisms are termed cell cycle checkpoints, and represent intrinsic signaling systems of cell cycle control. The checkpoint mechanisms also respond to external signals so that arrest may occur in cases of nutrient deprivation or growth factor withdrawal. The numerous checkpoints in the cell cycle, which are clustered in two major groups, those occurring at G1 and regulating entry into the S phase, and those occurring at G2 and regulating entry into the M phase, figure 2.2. This clustering suggests that intrinsic and extrinsic signals may funnel into common components of cell cycle regulation. Additional checkpoints ensure the orderly and dependent series of events which comprise mitosis. Different organisms attach varying degrees of significance to the G1 and G2 checkpoints, reflecting the stage at which a cell receives input from the environment. The checkpoint is predominant in the budding yeast Saccharomyces cerevisiae, where it is called start, and in animal cells where it is called the restriction point or commitment point. The yeast assesses nutrient availability in the presence of mating pheromones during G1, whereas animal cells respond to the presence of growth factors. Cell kingdoms will arrest at this checkpoint if the environment is unsuitable for growth, but once past it, they are committed to a round of DNA replication and mitosis regardless of their environment. In the fish and yeast Schizosaccharomyces pomba, the environment is monitored at the G2 checkpoint, and under satisfactory conditions the cell will undergo mitosis, division in the next round of DNA replication before checking again. The advantage of pausing at G2 rather than G1 for the haploid yeast cells reflects the presence of two copies of the genome at GP, allowing any damage to DNA to be repaired by recombination. Studying cell cycle regulation. Two complementary approaches have been used to characterize and isolate the regulatory components of the cell cycle. In the heterokaryon approach, nuclei at different stages of the chromosome cycle are joined in a common cytoplasm and their behavior observed. Cultured mammalian cells and amphibian eggs have been used for these experiments. The results of fusing cultured fibroblasts synchronized at different cell cycle stages are shown in Table 2.2. The ability of M-phase cells to induce mitosis in any interphase nucleus provided early evidence for the existence of an M-phase promoting factor. Similar results were obtained in Xenopus nuclear transplantation studies. Interphase nuclei formed spindles when injected into eggs arrested at the metaphase of meiosis I, and cytoplasm from these eggs could induce meiosis in oocytes arrested at G2. The large size of the eggs was exploited to purify the substance responsible, which was called maturation-promoting factor. Further, that maturation-promoting factor could also induce mitosis in somatic cells, and was in fact identical to M-phase promoting factor, which shared the same acronym MPF. The second approach has been to exploit the versatility of yeast genetics to isolate conditional mutants for cell cycle functions. Numerous CDC mutants, cell division cycle, have been identified which are blocked at various stages of the cell cycle, yet continue to grow. A second class of so-called WE mutants allows precocious transition of cell size checkpoints, and are smaller than wild-type cells. The genes identified from CDC mutants are not specific cell cycle regulators, but control processes such as DNA replication, repair and mating, upon which the progress of the cell cycle depends. However, a number of CDC genes appear to play a direct role in the regulation of the cell cycle, as discussed in the following section. Satisfyingly, the biochemical analysis of MPF has shown that both approaches have converged on the same small group of molecules. 2.2. The molecular basis of cell cycle regulation: cyclins and cyclin-dependent kinases, CDK. The sequential stages of the cell cycle reflect alternative states of phosphorylation for key proteins which mediate the different cell cycle events. The cell cycle transitions represent switches in those phosphorylation states. The G1S transition involves the phosphorylation of proteins required for DNA replication, whilst the G2M transition involves the phosphorylation of proteins required for mitosis. The basis of cell cycle regulation is a family of protein kinases which phosphorylate these target proteins and hence coordinate the different activities required for each transition. Of protein kinases in cell cycle control was revealed when analysis of S. cerevisiae CDC mutants blocked at start identified the product of the CDC28 gene, a 34Kd protein kinase, as the principal regulator of the G1S transition. The C2 gene, which played an equally important role in the G2M transition in S. pomba, was found to encode a homologous protein kinase. Genes encoding similar kinases were subsequently isolated from vertebrates, and these could restore wild-type cell cycle function to yeast CDC mutants. Significantly, the Xenopus homologue of CDC28, CDC2 was found to be a component of MPF. The kinases were found to be present constitutively in the nucleus, but to control cell cycle transitions their activity would have to oscillate. An explanation for their periodic activity came from a study of sea urchin eggs, wherein were discovered a family of molecules whose synthesis and activity oscillated with the cell cycle. These molecules were termed cyclins and they were subsequently found in many other eukaryotes including yeast and vertebrates. The second component of MPF was found to be AB-type cyclin. The active MPF resides in the catalytic kinase subunit, but it is dependent upon the cyclin subunit, which introduces a conformational change in its partner to stimulate kinase activity. The cell kinases are thus described as cyclin-dependent kinases, CDKs, and function as CDK cyclin holoenzymes. This strategy of cell cycle regulation appears to be conserved throughout the eukaryotes. CDK cyclin diversity in the yeast and animal cell cycles. A number of potential CDK have been isolated from yeast, but only CDC28 in S. cerevisiae and CDC2 in S. pomba appear to be directly involved in the cell cycle, and are required for the GIS and the GIM transitions in both species. In animal cells, there is a greater diversity of CDKs. The first to be discovered, the P34CDC28, CDC2 component of MPF, appears to function specifically at the G2M transition. 10 or more further CDKs are present in animal cells. 5 of these are involved specifically in the early stages of the cell cycle. Diversity of cyclins is greater than that of CDKs, as different cyclins are synthesized at different stages of the cell cycle in both animals and yeast. There are at least 8 families of vertebrate cyclins designated AH. Since CDKs phosphorylate different targets at each cell cycle transition, cyclins are required not only for kinase activity, but also for substrate specificity. In animals, alternative cyclins may be differentially expressed in different cell types, which would facilitate the unique aspects of cell cycle control in distinct differentiated cells. There are generally 3 types of cyclin in all organisms: the G1 cyclins which regulate the GS transition, the S-phase cyclins which are required for DNA replication, and the M-phase cyclins which are required for mitosis. M-phase cyclins include the S-cerevisiae cyclins, the vertebrate A and B type cyclins, and the S-pomba cyclin Sig13. They are stable proteins but share a conserved motif called a destruction box, which is required for targeted ubiquitination resulting in degradation during mitosis. Other cyclins are inherently unstable because they carry a PES domain, and their roles are determined primarily by the transcriptional activity of their genes. All cyclins carry a conserved motif, the cyclin box, which is required for CDK binding, figure 2.3. The yeast and animal CDKs and cyclins involved in cell cycle regulation are summarized in table 2.3. Regulation of CDK cyclin activity. Transitions are characterized by bursts of CDK activity which cause sudden switches in the phosphorylation states of target proteins responsible for cell cycle events. Sudden spikes of activity are not regulated by cyclin synthesis and degradation alone, as cyclins accumulate gradually in the cell and only the meiotic cyclins are degraded by rapid, targeted proteolysis. 2 CDK cyclin systems are active in G1 of the mammalian cell cycle. The CDK2 cyclin E complex is required for the GIS transition. The CDKs and the D cyclins are responsible for interpreting growth factor signals for the environment, and act at the restriction point to channel the cell into either late G1 or GO. The mammalian CDK1 cyclin B complex is MPF, the vertebrate homologue of yeast CDC2, CDC the 28th of may be termed CDC2 or CDKL. CDK7H cyclin, which is CAK, the mammalian CDK-THRL61 kinase, is thought to be expressed constitutively. CDKs phosphorylate target proteins, but are themselves also regulated by phosphorylation. CD28 and CDC2 CDKs are phosphorylated on 2 key residues, TIR15 and THR161. THR-161 is required for kinase activity, whereas phosphorylated TYR15 is inhibitory and dominant to THR-161 phosphorylation. The principal determinant of CDK cyclin activity in yeast is thus the state of phosphorylation of TYR15, and some of the upstream regulatory components have been identified. S4D, WEE1 is a tyrosine kinase which phosphorylates CDC2 at TYR15 and thus inactivates it. WEE1 activity is antagonized by CDC25 phosphatase, which removes phosphate groups from the same substrate. WEE1 and CDC25 are themselves regulated by intrinsic and extrinsic signals, and this is believed to be the basis of the G2M checkpoint in this species. The data to proceed with mitosis or arrest in G2 thus reflects the relative levels of these opposing activities, and the regulatory networks which feed into this checkpoint are considered below.